Good afternoon. This presentation is about the impact of the gut microbiome on whole body health. Microorganisms, including bacteria, viruses, and fungi, live on the inside and outside of our body and are commonly known as the microbiome. Scientific research into the billions of gut microorganisms of the digestive system is continually developing to help us understand their influence on our general health. What is the level of evidence on gut microbiome? Recent evidence-based research provides us with a better understanding of the role and function of the gut microbiome. However, it must be stated that research is ongoing. Although the gut microbiome is associated with disease, there is no conclusive evidence that abnormal gut microbiota is the cause of disease. This presentation will report on the latest research to describe the role of the gut microbiome of the gastrointestinal tract, how the gut microbiome affects physical and mental health, what positive lifestyle and dietary factors influence the gut microbiome, and what negative lifestyle and dietary factors influence the gut microbiome. Describe the role of the gut microbiome of the GIT. General role of gut microbes. The majority of the gut microbiome live in the colon. Each individual has a unique signature of gut microorganisms with a basic function to consume nutrients and secrete molecules. These molecules can be ingested by our own body cells and may be beneficial or harmful to our general health. This important function demonstrates a symbiotic relationship between the gut microbiome, lifestyle, and our general health and well-being. Specific role of gut microbes. The following roles of the gut microbiota include the digestion of fiber, the development and maintenance of the immune system, maintains the gut lining and protects against disease, the development and maintenance of a healthy metabolic system, and may protect mental health. How these roles influence physical and mental health will be explained on the following slides. The role of the gut microbiome in influencing physical and mental health. Fiber, the immune system, disease prevention, and a healthy metabolic system. So let's look at various influences of our gut microbes on our physical and mental health. First of all, we know certain microbes like to digest fiber. These bacteria secrete short-chain fatty acids that feed the cells of the epithelial wall in the colon. Healthy epithelial cells support the immune system by preventing pathogens into the bloodstream. Furthermore, this also prevents low-grade inflammation that may lead to metabolic syndrome and may result in diseases such as obesity, type 2 diabetes, or cardiovascular disease. This evidence-based knowledge is supported by nutrition scientist Dr. Joanna McMillan, who explains the role of our gut microbes is to digest fiber and secrete short-chain fatty acids, which help feed gut cells, trains and keeps the immune system in balance, and reduces the risk of inflammation. Fiber, the immune system, disease prevention, and a healthy metabolic system. The role in the first 1,000 days. The first 1,000 days of a human life sets the foundation of gut microbiome and impacts physical and mental health. Environmental conditions determine the types of microbes gathered in the first 2 years of pre- and postnatal life and can provide a functional purpose for disease prevention and metabolism. A relationship exists between irregular gut microbiome and abnormal physical development during the first 3 years of life and can lead to a chronic disease later in life. The 2016 literature review into the gut also explains the function of the gut microbiome to protect against diseases. A disruption to the microbiome Bile homeostasis in the first 2 years of life can lead to many disease outcomes such as asthma, allergies, immune deficiencies, and obesity. For example, the use of antibiotics in the pre- or postnatal period destroys healthy microbes and increases the likelihood of disease including asthma, inflammatory bowel disease, and obesity. This literature review underlines an essential functional relationship between the type of gut microbiome collected in early life and their influence on physical health. How microbes can influence food choice, metabolism, and mental health. In Australia, 2 in 3 people over the age of 18 are overweight or obese. Individual food choice and risky weight gain has traditionally been seen as a lifestyle-related health issue. Recent insight into the gut microbiome provides compelling evidence of microbial influence on our dietary behaviour. Research findings indicate gut microbes may manipulate eating behaviour using a variety of strategies. These include 1. Influencing reward and satiety pathways such as the vagus nerve to generate cravings for specific foods that help to flourish certain microbes. Specific microbes can create adrenergic neurochemicals that may stimulate receptors of the enteric nervous system, which further stimulates the vagus nerve to drive a pattern of excessive food consumption. And 2, producing neurotoxins to change receptors and mood, therefore influencing the eating of specific foods. Certain pathogens have been found to increase anxiety levels and may influence poor eating decisions. A 2019 breakthrough study of 1,063 people into gut bacteria and depression provides a relationship between microbiota and depression. 2 sets of bacteria were lacking in people with depression, whilst 2 types of bacteria that strengthen the epithelial cells and the immune system were only consistent for people with a higher quality of life. This indicates an association, however does not provide evidence that gut microbes cause depression or affects an individual's quality of life. Positive and negative factors influencing the gut microbiome. Early life. Evidence has developed over the last decade underpinning the relationship between a dysbiosis in gut microbiome and autoimmune disease. This relationship is more prominent in early life when the development of gut microbes are largely determined by specific factors. A literature review study explains the healthy development of an infant's gut microbiome occurs in response to the following key factors. The type of birth process. An infant delivered vaginally receives essential bacteria that inoculates against disease. An infant delivered via C-section does not receive this bacteria. The level of breastfeeding. Breast milk contains oligosaccharides, which are consumed by bifidobacteria. Short-chain fatty acids are produced and are used to strengthen the cells of the epithelium of the colon and protects us against infection and inflammation. These bacteria can also crowd out less favorable bacteria. Antibiotics. The use of antibiotics is not recommended during pregnancy as it results in the flora of the gut being destroyed, which has been associated with the development of asthma, type 2 diabetes, and inflammatory bowel disease. Host genetics. A dysbiosis of the adult gut microbiome may be genetically inherited. Host infections. A vaginal infection can result in bacteria entering the uterus and fetus. Probiotics. Consumption of probiotics may reduce gut infections. If these environmental conditions are hindered, for example, A balance of unhealthy microbes may produce secretions in the infant's intestinal tract that can trigger an autoimmune or inflammatory response, leading to chronic disease. Gut diversity and the Western diet. Research into what is a gold standard of gut microbiome is inconclusive. But what we do know from studies undertaken with various populations is that cultures with a higher diversity of gut microbiome present with less chronic disease. Catherine Lozapone, a leading microbiologist who is working on the American Gut Project, explains how the gut of people living in rural areas in Africa and Venezuela are more diverse than the people of America and Europe. Lozapone highlights how these rural people consume more plant-based foods, which correlates with less chronic health disorders such as allergies, asthma, type 2 diabetes, and cardiovascular disease. The Western diet concentrates more red meat and refined carbohydrates, whilst providing for less fruits and vegetables compared with many non-Western diets. Leading microbiologists of the American Gut Project explain how a lack of fiber decreases gut microbe diversity, increases the permeability of the lining, which may be the cause of higher rates of type 2 diabetes, cardiovascular disease, and colon cancer. Gut diversity can be increased from eating a range of vegetables, fruits, grains, and nuts. A balanced diet. Fibre is particularly important for the health of the gut microbiome. The Australian Institute of Health and Welfare report Australians from the age of 9 years of age did not generally meet daily fibre intake of 24 to 30 grams for males and 20 to 25 grams for females. A well-balanced diet with adequate whole plants and grains will provide the recommended dietary intake of fibre for the general population. According to Food Standards Australia and New Zealand, the types of fibre that support a healthy colon are soluble fibre, which is found in fruit, vegetables, oat bran, barley and legumes. Insoluble fibre, which is found in bran, fruit and vegetable skins, whole grain foods. And resistant starch, which is unprocessed cereals and grains, unripe bananas, cool potato and rice. Current research into whole grains have linked cereal fibre to a reduction to the risk of colon cancer, type 2 diabetes, obesity and cardiovascular disease. Furthermore, a low intake diet of whole grains is associated with the most deaths and DALYs. Other positive influences. Most of our microbiome is found in the digestive system, so what we consume has a direct relationship with the health of our body and mind. Harvard Medical School provide the following considerations to maintain a healthy gut microbiome. Choose a Mediterranean diet, a diet consisting of a variety of fruit and vegetables, green leafy vegetables, olive oil, tomatoes and fish. These foods are known to protect against inflammation. Maintain good hygiene. Various hygienic strategies are recommended to minimize bad bacteria. These include wash hands after using the toilet and before preparing meals, and brush and floss your teeth after a meal to protect the biofilm from bad bacteria. Eggs, meat and fish. Microba, a world-leading company in the analysis of the gut microbiome, reports excessive levels of hydrogen sulfide is produced when large amounts of eggs, meat or fish are consumed. Too much hydrogen sulfide negatively impacts the epithelial wall and promotes inflammation. However, small amounts can be a positive influence and can be used by epithelial cells for energy. Thank you very much for your time.